This is Thomas Maiwald, currently from Harvard Medical School, and I will speak about dynamic modeling of biological systems.
Good afternoon to Germany. Thank you, Monika and Thomas Zimmermann, for your commitment to support basic research. I worked during my PhD thesis in the group of Jens Timmer here in Freiburg, and now I'm working in Peter Sorger's group here in Boston at the Harvard Medical School. My thesis deals with the dynamical modeling of biological systems. I was involved in developing experimental and mathematical methods to improve the data quality, mathematical modeling in general, and to design optimal experiments. I just realized that you cannot see my head of the slide, so I will just tell you what is written there with this. All concepts that were developed were implemented into software so that other researchers can benefit from our experience. And the software was used in several signal construction pathway modeling projects. For example, to do better, as Stefan just explained about, or the EVO receptor that was presented by Lena Becker yesterday. Today, I would like to focus on 3 projects: the data quality of Western blotting, mathematical modeling, and the protest software. When I started PhD, I worked together with Marcel Schilling from the lab of Ursula Klingmüller in Heidelberg on modeling the MAP-KNS pathway. And we were curious whether we could find oscillations in the system, because they were suggested by Professor Kolodenko as one possible state of the dynamics. Well, here we have some kind of oscillation, but what I have not yet shown to you is the legend, what we've actually measured, and the legend for the x-axis. Measured is the total Earth concentration, which should actually be constant for this experiment and not oscillate or change at all. And the x-axis is the gel length. So instead of time oscillation, an oscillation in time, we have an oscillation over the gel. That means the gel, the Western blotting gel, tends to correlate neighbored lanes and give them either increased or decreased value. This could have strong effects on the modeling if we assume that we make a time course experiment and load each time point into consecutive gel lanes. That means the first time point in the first slot and so on. Then we would have an oscillation because there's a systematic problem with this measurement technique. And in reality, the data is maybe even constant. Marcel and I tried to figure out the source of this problem, and to summarize this, it's not the reprobing with new antibody or the stripping process of the gel, it's the blotting step itself. Here we have again total Earth measurements on 2 gels, and as you can see, for the one gel we have a big sine wave, and for the other gel we have 2 smaller sine waves, and it's very important to not let these furious dynamics get into the modeling. We suggest a 3-step process to tackle the problem. The first, the easiest and the most efficient one is to just use a randomized gel loading. It means we do not use the consecutive loading of the gel, but we, in a random manner, and if we then reorder all data points, as shown here, and then average neighboring time points, then we get the constant line that we expect for the total Earth concentration. The second step is to use normalizer proteins, and the third would be to merge data from several genes. And to give you the answer for the oscillatory question, well, with this approach we have not found any oscillations in the MAP-KNS pathway. Now that we have suitable ways to get quantitative data, we can start mathematical modeling. This means we want to transfer a biological hypothesis into a formalized mathematical model, here as a biochemical reaction network, which corresponds to a certain set of differential equations. The network contains unknown parameters, which are rate constants usually in the system, and we are now trying to calibrate these parameters until we can explain measurements as best as possible by the model generated. The process of parameter calibration is shown here for two parameters, starting from an initial guess, doing optimization, the parameter values change, and finally we end at a certain point in parameter space. The question arises, how strong does this process depend on the initial guess? So if we apply fit sequence, where we start from different points, usually we end up not at the same point, but rather in some kind of manifold. We see this if we repeat the fit sequence very often. All these points on this manifold, this line, correspond to the same fit quality, so we cannot decide which one of the parameter combination is now better or worse than the other. The problem is called non-identifiability. One way to resolve the problem is to use data from several experiments at the same time to apply multi-experiment fitting. Here's one example where we use a synthetic model from the process pathway. If we use only one experiment and fit the parameters, we see a very broad distribution for each parameter. If we now combine this single experiment with 3 further experiments, the identifiability problem is resolved for most of the parameters. Multi-experiment fitting is not also useful to resolve this issue of identifiability, but also to discriminate competing model hypotheses. For this, I would like to switch to the program photoscreen. And we are now going to compare 3 different receptor models.